Welcome, everybody, to the Playing Games podcast. And today I have a very special guest, one of the leading researchers in muscular fatigue, Dr. Håkan Westerblad. Am I pronouncing that right?
Yeah, almost. Håkan Westerblad in Swedish, but that's very difficult to say if you're not from Sweden.
Thank you, Dr. Westerblad, for joining us today. Why can't we, let's start off with your introduction a little bit on in the field of exercise physiology and fatigue and what kind of brought you here where you are today, just for people who may not be familiar with you or your work so much, just to get a small introduction.
As a small introduction to me, my background is I have a medical degree, but I only practiced the medicine for 6 months, something like that. So in 1984, I started my research on muscle fatigue, recovery, and so on, and with a focus on the single muscle fibers. In those days, everyone that worked on this kind of cellular mechanism used frog fibers, and it was supposedly impossible to do it on mammalian fibers. But I spent the summer early on to see if it was really impossible, and it was possible. So since 1987 or something like that, the main focus has been fatigue, recovery, training, and so on, with the focus on single mammalian muscle fibers. Starting with mouse, then rat, some years ago also human muscle fibers, or muscle cells if you want to call them that.
Has it been cool to see the transition in technology from when you first started to where you are now, and how much more accessible it is to look at fatigue in certain ways?
Yeah, that's been quite a lot of development, yes. When we started, calcium is a central thing in muscle fibers. So calcium is stored inside the muscle cells, and then when the nervous system tells the muscle fiber to contract, it sends a signal, so calcium comes out from the stores into the cytoplasm and activate the motors, and the muscle fiber contracts. So calcium was known to be a very central theme, but the methods to measure it were rather complicated to use when I started. So to begin with, we measured force and during repeated contractions, and look at the basic force properties during fatiguing stimulation. But then around 1990, something like that, then it became more commonplace to use fluorescent indicators. And with those, you can measure calcium and magnesium and pH and all sorts of things. So that was a great technology development. And then, of course, when you work with mice, then also came the molecular era, so to speak, with all the molecular methods, including that you could increase or decrease the protein expression in the muscle fibers and see what's happening then.
Awesome. So I kind of wanted to go over specifically a recent paper that you published. I think it was in 2024, where you kind of went over a lot of the literature on fatigue and how some things are overstated. But we can start off with peripheral fatigue, kind of like what it is, how you define it, and I guess what it includes. And then we can get into like some of the nitty gritty of the mechanisms that cause peripheral fatigue.
Yeah, so peripheral fatigue in my mind is when you do perform exercise and so on, then of course it starts with the brain sending down signals, telling the muscle fibers to contract, and they contract, they produce force or velocity or power, and so peripheral fatigue, in my mind, it's sort of an acute experience. With acute, I mean, it could occur in minutes or up to a few hours, not something that takes weeks to develop. So the kind of peripheral fatigue we're looking at comes rather abruptly and recovers to some extent, rather fast too, but could be long-lasting. So at any rate, so that is that the muscle fibers, despite if they have optimal or maximum activation, they become weaker and slower. And that is what the fatigue is all about. And if they become weaker and slower, that also means that the power goes down. Power is force times velocity.
And do you know some popular or most common research methods to look at peripheral fatigue that you currently use?
And so what we have been using over the years is isolated single muscle fibers. So you dissect them out from the whole muscle then. It's a bit tricky. You need a special kind of microscope and also some skill. You do it with a small scissor in one hand, scissor that normally used for eye surgery, and small forceps in the other one. They're the ones that the people that repair the watches used to use when they still repair the watches and not just throw them in the bin and took a new one. So you dissect out a single muscle fiber, and then you can do that muscle fiber sort of doesn't really understand that it's no longer sitting in the muscle or in the animal. So you can stimulate it with electrical currents. You mimic the nervous system in that way, and then it produces everything that it used to produce to contract when activated this way. So the reason as to why we do micro dissection is that then we also get the tendons intact on both sides. It's much easier to do skin fiber preparations. You can do that from, for instance, from human biopsies. So you take out a piece of muscle, you can treat it in different ways, so you get single muscle fibers, but then they don't have any tendons. So then you can't measure force the normal way or shorten velocity. You can measure a lot of other things, but it's not really the real thing anymore.
So with peripheral fatigue, there are, from what it seems, a lot of mechanisms going on there. And we could kind of start with the decrease, or all of them seem to have a decrease in force generating capacity. We kind of start with the first one, often seems to be the main one, which is reduced calcium availability or from the sarcoplasm. If you kind of want to go over that one and some mechanisms for it, because it also seems like all of these tend to intertwine at some point or have an interplay with each other.
Yeah, so an important mechanism there is that, like I said, that when a muscle is going to contract, what's happening inside the muscle fibers is first that calcium is being released from the internal stores, and that activates the motors and the muscle contracts. So that's great. The problem there from a fatigue perspective is that when the muscle fiber contracts, you have the cross bridges, the motors, they're going then to do the contraction, but they consume energy in the form of ATP. And then also you need to be able to relax all the time. So as soon as there is no action potential triggering calcium release, or rather calcium is being pumped back into the stores again. For it to be outside of the stores, it has to be released all the time. And as soon as it's released, the pumps in the sarcoplasmic reticulum tries to pump it back. And they also consume energy in the form of ATP. So you have two major energy consuming things going on as soon as you start to contract. And you definitely have to make sure that the ATP, the immediate energy store, doesn't, it mustn't go down to low levels, because then the muscle goes into rigor and dies. So you need to have some protection mechanism so that if the energy consumption is too high, you need to slow down the energy consumption. And then a perfect way to do that is to reduce the release of calcium from the sarcoplasmic reticulum. Because if you do, then you don't activate as many cross bridges anymore, energy consumption goes down, and you don't have to pump calcium back into the sarcoplasmic reticulum, which also means then that the energy production consumption goes down. I'm sorry, it's a small dog here that wants to have attention. Please go.
So
So that is so then one can say that the fatigue mechanism you talked about, the reduction in calcium release, is there so that it should be impossible for the muscle fibers to kill themselves. And there are lots of mechanisms then. I mean, this is essential for life, one could say. So there are many ways to reduce the SR calcium release because it mustn't go wrong.
So it's like it's the fatigue is kind of like a protective mechanism in a way, right? For the calcium to be reduced because it looks like calcium is like a double-edged sword, right? It's needed for muscle contraction, but too much of it can start maybe causing problems.
Yeah, it will definitely cause problems, yes.
Okay. So with the reduced calcium, I think the next I guess we could get into the mechanisms because I think these all kind of play a role with reducing calcium. Is acidosis or hydrogen ions specifically? I know they have some contention around them, but it seems you pointed out that they could be overstated in the fatigue literature. Um,
Yeah, if we talk about the sarcoplasmic reticulum calcium release, then I think basically everyone agrees that when you do intense exercise, then you get lactic acid and hydrogen ion accumulation, but they don't do anything really to SR, sarcoplasmic reticulum calcium release. They can do other things, but maybe we'll come back to that. But I think everyone agrees upon that they are not a cause of reduced release of calcium.
the point with lactic acid, I know people kind of go back and forth whether it's lactic acid or lactate. Do you think it really matters or anything like that? Or
No. I mean, it isn't lactic acid from a chemical point of view because lactic acid is a strong acid. So basically, all of the lactic acid would be in the form of lactate ions and hydrogen ions, where lactate ions as such don't really do a lot. But they're easy to measure, so from that perspective, they're interesting. They're exported out of the fiber during exercise, and you can measure them with an easy blood test.
so while they don't really contribute to reduced calcium sensitivity, or to the release of calcium, they can contribute to other things, right? Like as reduced shortening velocity, right?
Yes, so there is the point where hydrogen ions in acute fatigue can be of importance. They can both make the cross bridges weaker, which means that the muscle fiber becomes weaker, and they can also slow down the cycling rate so that the muscle fiber becomes slower. And then there is a debate then on how much. Is this the most important component in this effect, or is it not as important? So there are intact fiber data and some other basic data to indicate that they aren't very important. They have some importance, but not an awful lot. And then you have some of the skinned fiber experiments that sort of mentioned the easier way to get single fibers you can get from biopsies and so on. Some of which those experiments indicate that they are enormously important.
So, do you think, or how exactly does acidosis reduce shortening velocity, and how can it reduce force if it does so even by a little bit?
The complicated answer, you should really talk to one of the sort of cross-bridge experiments. You mentioned the recent paper that I wrote together with Ned DeBold, who is sort of the myofibrillar expert on fatigue. So in that paper, there is a lot of possible mechanisms as to how hydrogen does it, all of which I could say are very complicated, even for me, who have worked with muscle fatigue properties for 40 years or something like that. So in principle, they to make the cross bridges slower, it's actually that in some way each cycle where they put in an ATP and out comes ADP and phosphate, it goes slower. And there are various ways this could happen. And regarding the decline in force, it's that when they pull, or I think most of the time one believes that there are fewer cross bridges pulling when you have a metabolic problem like hydrolytes and so on. So if the myosin head, the cross bridges pull on the rope, the actin filament, there are fewer pullers at a time, and then force goes down.
So they don't reduce force through reducing the release of calcium, but more so just messing with like the motors in the muscle, right?
Just
Having a direct effect on the motors in the muscle, I believe.
yeah, yes, it's direct, but not simple.
Yeah, it always comes up to me that fatigue is always like, oh, well, you have this metabolite that's very simply just reducing force. But I think when we get into like how it does it, in the paper, you guys go over a variety of research that still is, we remain somewhat unclear on certain things, just showing how much more we still need to look into. But another popular metabolite is inorganic phosphate, and that one seems to get all the attention for reducing force. I'm not sure if it reduces speed so much, right?
No, that apparently the speed is not much affected by phosphate ions. So the cross bridges cycle at the same rate as they do in, or they say cycle at the same rate in low and high phosphate.
So how does phosphate? Do you know how phosphate or inorganic phosphate causes a reduction in muscle force? I think that one does reduce the release of calcium ions.
Yes, so phosphate can reduce both the release of calcium from the sarcoplasmic reticulum and reduce the force that the cross bridges produce. So during fatigue, what happens first after a few contractions? You can only do whatever. Say pull-ups, you can do 10, but you can't do 11 because now you have a muscle fatigue. And then most likely the calcium release is not the problem yet. It's actually the motors can't produce as much force as before. And with phosphate, that's because fewer motors. At each time point, there are fewer motors that sit on the actin rope and pulls, and therefore force goes down. So then force goes down both because you have fewer, but also that you need more calcium to activate the machinery when phosphate increases.
But how can you tell it was from a reduction in motors and not from the release in calcium?
The way we have distinguished that over the years is with caffeine, which is the basic tool. So if you add caffeine to a muscle fiber, you get release of calcium from the sarcoplasmic reticulum without any action potential, or it potentiates the release. But you need, if you want to do it on your own, so to speak, what is it, something like 500 cups of coffee or something like that. So
Oh,
it's
wow.
pharmacological, or experimental doses. You can never reach that state in real life. But then calcium comes loose. So if you have a fatigued situation with reduced calcium release, and then you put on caffeine, then calcium release briefly goes up to a normal level. And then you can see how much force you have. And then normally the force is decreased by 15-20% or something like that, which is then the cross-bridge part of fatigue. And the rest is then the calcium release problem.
So I know people like having an answer when they're in the gym, and let's say they're doing like a bicep curl for around 8 reps, and then, or even high reps, and then they start slowing down and they fail the set. is that could be because mainly of the reduced calcium being sent out, or is that more so with inorganic phosphate messing with the motors, or could it be both, a mixture of both? It's hard to tell which one is more than the other.
Yeah, it depends, I guess, how many repetitions you're doing, and then also if you're close to maximum, if you can only do a few, so whatever you do, you say you need 85% of your maximum force or so, then you can only do a few. Then I think the calcium release part is not really going to develop. It will be more that the motors are weaker, because that comes first. But if you
So
can do 50 repetitions, then I'm pretty sure that the calcium component comes in too.
let's see. So the higher reps that we do, like let's say from going from like 1 to 30, that's when we start messing with some calcium release. But if we're doing like some lighter or lower rep ranges, it could be more just motor ability of the muscle happening.
Yeah, I think so. As a basic concept, so to speak. The
And is there
metabolic part, when you really use heavy weights and few repetitions, then the metabolic demand is not that big, I imagine. I'm far from an expert on this kind of exercise in real life, so to speak, but I imagine it's tougher on the cardiovascular system and so on when you do many repetitions than when you only do a few.
Do you think there's ways to measure that currently, or when it comes to like a resistance gym setting, it's kind of very difficult to look at fatigue?
You could, with electrical stimulation, of course, you could test this and see if it has gone down. Uh, to do something easy, no, I don't think it's really practical. But in principle, you could measure if it's calcium release or if it's the myofibrils.
That's pretty
But you
cool.
clearly can't measure calcium in a person in a gym. There's no way that you can measure cytosolic calcium release or calcium change.
Well, the next one from inorganic phosphate ends up moving to reduced ATP and increased magnesium, right? Do you know what how those mechanisms are, or what kind of what's happening there?
So now I think now we're talking about sarcoplasmic reticulum calcium release, are we? I guess
For
you jumped to that.
yeah. So we went from acidosis, inorganic phosphate, um,
Yeah.
and then the next one is reduced ATP and increased magnesium. That was another mechanism that was in the paper.
Yeah. Okay. Yes. So phosphate. When phosphate increases, it can reduce sarcoplasmic reticulum calcium release. And what seems to be the major mechanism there is that phosphate comes when your metabolic demand is higher than what you can get from aerobic metabolism. And then you start to form lactic acid that we can forget about for the time being. But then you also break down phosphocreatine to creatine and phosphate ions. So the phosphate ions accumulate in the cytoplasm, and due to some mechanism that's not perfectly clear, some of these phosphate ions move into the sarcoplasmic reticulum. And in that store for calcium, you have a high calcium concentration. So if you have a lot of phosphate and a lot of calcium ions in the same place, you can get precipitation.
I think you cut off a little bit there. Sorry, could you go back a little bit
You go
before
back. Okay, so if you
the
have a lot
precipitation? It
of
was like
phosphate ions in the cytosol, some of them can enter the sarcoplasmic reticulum, the calcium store. So when then they enter the sarcoplasmic reticulum, you will have a lot of calcium ions and a lot of phosphate ions, which means that you can have precipitation.
Hmm.
So in that way, you bind the calcium so it can't be released anymore. And with severe fatigue, you can bind more and more of the calcium so it can't be released. So that's the phosphate mechanism for reducing SR calcium release. But then you said ATP and magnesium, and they are at another level. And that is when a muscle fiber is activated, it starts with an action potential traveling in the surface of the muscle fiber. And that activates calcium release channels in the sarcoplasmic reticulum. They are called the ryanodine receptors. And the ryanodine receptors, they are sensitive to ATP and magnesium. So if ATP goes down or magnesium goes up, then you inhibit the ryanodine receptors, meaning that you get less calcium release. And the interesting aspect there is that the energy stores, the ATP, is not really ATP, it's magnesium ATP. And when the store is depleted, then ATP goes down and magnesium increases at the same time. And both of them tend then to inhibit the release of calcium from the sarcoplasmic reticulum.
Does that have to be like a depleted ATP, or as soon as ATP starts going lower, then magnesium can start increasing? And just because it's not depleted doesn't mean it's not.
Yeah, it's far from depleted. It mustn't get depleted because then the cell dies. So it starts to decline already when the ATP is down a bit. And that also, if you do the most severe fatiguing exercise you can think of, and then you look in the muscle fibers, if the ATP, the energy store, has declined, and most of the time it has not really. Maybe if it's 6 millimolar in the rested state, in severe fatigue it might be 4 or something like that. There are severe cases when it's been down to 2 millimolar, but basically the ATP stores remain. They can't go down. If they go down below 1 millimolar or down to 100 micromolar or something like that, then you will have a dead cell. So that mustn't happen.
Yeah. So when we talk about fatigue, it doesn't just have to be ATP depletion. It could just be a decline.
Yeah,
A slight decline of ATP.
it's never ATP depletion.
And so with inorganic phosphate, a little bit, just to go back, that can enter the sarcoplasmic reticulum, which could start affecting the release of calcium release alongside with the motor, right? The motors in the muscle. And with reduced ATP, it's the reduction or the decline in ATP, even if it's slightly, and that can start to, or magnesium starts to increase as soon as that's happening simultaneously. And how does that one reduce fatigue directly again?
So that causes fatigue by inhibiting the sarcoplasmic reticulum calcium channels,
Okay.
the ryanodine receptors. So they aren't as effective. So when the action potential comes, they don't release 100% or open up 100%. They open up 90%, 80%, 70% maybe.
So that one doesn't mess with the motors of the muscle. That one's more so with just the release of calcium, right?
Yeah, I would say so. ATP, of course, mess up with the cross bridges too, but you need big changes there. And it becomes maybe a bit too complicated.
And we don't really see big change.
If you have when ATP is borderline or the energy buffer is borderline, then you get when you do exercise or contract the muscles, you get increase in ADP. So ATP, adenosine triphosphate breaks down to adenosine diphosphate. And the diphosphate version can slow down the cross bridges and affect the cross bridge cycling. Whereas ATP has to go down to really low levels before something happens.
Okay. And well, I mean, I think this one's a little related to that too, because the next one is glycogen. And glycogen's also a reduction in glycogen could also cause fatigue, right?
And basically, not directly. There are some suggestions that it could do it directly, but the glycogen is the carbohydrate stored in muscle fibers. So it's there to provide energy via aerobic and anaerobic metabolism. So when the glycogen store becomes depleted, low, then you don't have enough energy around. And then all the fatigue mechanisms start because of a lack of energy. So again, to prevent disaster from happening, safety mechanism. So glycogen stores goes down, and that will then inhibit SR calcium release, and also it could decrease the function or impair the function of the cross bridges.
But that one, like you said, is indirectly because it's more so that it's the reduction in energy that prevents everything else from going, everything that affects it directly from going on.
Yeah, I think it's rather universal agreement upon that. There
Okay.
are some reports, but I think they haven't really gone anywhere. That it is a structural part in it too to make it complicated. But basically, it's the same metabolic. So all of the fatigue mechanisms, one could say, are metabolically related. They're due to the fact that the contracting muscle consumes 100 times more energy than the resting muscle, basically. We were saying that that causes the same metabolic problems, but after some time. So for the glycogen depletion to be a problem, then you do need to do exercise at a slightly lower level. So if you say the fatigue at the end of a 400-meter sprint, it's not going to be glycogen depletion. Whereas if in a half marathon, for instance, it could surely be glycogen depletion.
But even if the glycogen is not fully depleted and it's like still slightly decreasing, that could also affect fatigue, right? Or it has to be depleted.
Then, then we enter yet another source of complication is that glycogen is stored in several places in muscle fibers. And some places aren't really involved directly in exercise and so on. They are there to do other things. So if you take out a piece of muscle and measure glycogen, it's never really depleted. But if you look carefully with the electron microscope or so you can see at the micrometer scale, look at the glycogen, then it can be depleted. For instance, the glycogen required for calcium release from the sarcoplasmic reticulum. So that can be depleted despite the fact that in the rest of the muscle fiber there's still glycogen available.
So glycogen depletion is not uniform across muscle, right?
No, it's not uniform across different muscle fibers, and it's not uniform within the individual muscle fibers either.
Oh, okay. So I thought it was just like some fibers can be depleted while others are good or not
Yeah,
depleted.
yeah, that's fine. But then if you then go in and look at the depleted fibers, they have differences. Could be parts of the depleted muscle fiber where you still have quite a lot of glycogen, and others when it's empty. When we've done experiments too on single fibers where it's easy, so to speak, then it appears that glycogen goes down with exercise. Then you reach sort of a critical level. And at the critical level, then you see the decrease in sarcoplasmic reticulum calcium release and so on.
So you did say with a 100-meter sprint, it may not be glycogen depletion, but
No, definitely not.
there could still be depletion of glycogen with certain fibers or within certain fibers of the muscle, while others are fully.
No, not for that short period. Because then you get the metabolic problem during a 100-meter sprint, so to speak, is that the maximum speed of fat oxidation, glycogen or carbohydrate usage and phosphocreatine usage, it's exceeded. So you consume more energy than you can produce, even on the very, very short time scale. And then you have fatigue.
Because I know with resistance training, um, it's not really something that will deplete glycogen, but there are some. I did see some papers showing like some fibers might, like the type 2 fibers that we really use a lot for something heavy, like some of those might be depleted, but as a whole, the muscle is not really depleted of glycogen. But it's like certain fibers within the muscle could be depleted.
Yeah, I imagine so. If you use heavy weights, so few repetitions, which means, and heavy weights, which means that you can activate the muscle fibers that you normally don't activate. You activate the fastest, most explosive muscle fibers, and they are useless in metabolism, so to speak. They are there to contract every now and then, but if you use them uh, more frequently during a really heavy uh, exercise protocol in the gym, then it might be that they are depleted to some extent, at least. Whereas the
Right.
others, it's sort of a normal day at work, don't really care.
What about with like higher rep ranges, probably, or would that be more to like sprinting where it's very like metabolite buildup?
Yeah, it could be, yes, yes. And also, if you do more reps, that means that you have lower weights, so to speak, which then also means that you're probably not going to activate the strongest and fastest muscle fibers anymore because you can do it without them.
Yeah, and if
And then you activate the metabolically more durable fibers.
and then the type 2 fibers are activated at the end for like a short period of time compared to like heavy weights where they're activated pretty much throughout the entire
Yeah,
set.
yeah, exactly. I mean,
Okay.
the really fast, strong type 2 fibers with many repetitions, they are probably activated not until the other fibers are severely fatigued. So then to do one more rep, so to speak, you have to activate those fibers too.
So the next, I think there are two more mechanisms that we mentioned that are mentioned in the paper, and the next one is impaired sarcolemma excitability. Maybe if you could elaborate on what that is a little bit and its contribution to fatigue and force loss.
Yeah, so that is sort of the first step in the activation process. And that is when the nervous system sends down an action potential to activate a muscle fiber. It's then transmitted to the muscle fiber, where you get a new action potential traveling along the surface of the muscle fiber. And a classical model of fatigue is that these action potentials traveling on the muscle fibers, that they become the amplitude goes down, they become slower, and that is a cause of fatigue. Meaning that the very first step in the peripheral fatigue sequence is problematic. And there are some indications that this could be a problem, and there are quite a lot of data suggesting that it's not very important. And the nervous system, in my mind, is clever, so it's never going to send down action potentials at a higher frequency than the muscle fibers can cope with. So it is definitely possible that you have problems with the action potential propagation, but it seems not to be a very common cause of fatigue. It used to be more common cause of fatigue, so to speak, before the era of calcium measurements and uh, those kind of experiments, because then one could measure action potentials, and then one could stimulate the muscle fibers of muscle at a supraphysiological frequency, say 200 hertz continuously, 200 action potentials per second continuously, whereas the nervous system will in humans go down to 10, 15 per second. So if you overdo it, the activation that way, then clearly you have a problem with action potentials. But in real life, it's not going to happen.
And not, it's very rare for it to happen in any scenario, whether it's endurance or resistance training or anything like that. Or
I would say so, yes.
but the idea would be that that one would also reduce calcium release, right?
Yeah,
Because
yeah. So then there's the electrical signal that is going to open the sarcoplasmic reticulum calcium release channels is weaker. And then the opening of the channel is reduced. So
could
the
that
opening of the channel could be reduced by magnesium ATP acting on the channel, but it could also be reduced because the electrical activation is impaired.
um, could that also, well, actually, with magnesium, would magnesium also impact anything, or is it mostly just the reduction in ATP and the magnesium is just the increase that's happening?
Yeah, I would say so. I mean, of course, magnesium and calcium, they normally compete with each other, so magnesium can do other things too. But I would say that there are, as far as I reckon, that there are no convincing evidence that the magnesium would go in and do something else there in the system.
So it's just increasing and competing with decreasing calcium, I guess, right?
Yeah, it's together with ATP, it could cause a decrease in calcium release, yes.
And I mean, with the sarcolemmic excitability, could it be influenced by damage per se? Let's say if muscle damage occurred, or if there's like sarcomere disruption, could that possibly affect the sarcolemmic excitability the next day if you're still not fully recovered?
Yeah, surely. I mean, if the sarcolemma is not intact, then the membrane potential, which is totally essential for the muscle fibers and all cells, then that goes down. And then, of course, the action potentials don't work anymore. So if you have damage to a muscle fiber and it gets depolarized, it can't contract anymore. Then it has to be repaired one way or another.
But could it still be intact, maybe a little disrupted? But I mean, from what I've seen, the sarcolemma can be a little, it doesn't have to tear, it could just be a little disrupted, but still be fully intact. All right.
Yes, yes. And there, I mean, in principle, it could be that then the action potentials have problem passing that area, uh, hypothetically. And if they have, then they won't activate calcium release in that particular area. But I would say that I don't think it's a very common mechanism. But of course, in various types of injuries or something like that, it might happen.
So I mean, the last mechanism would be reactive oxygen and nitrogen species. Uh, what's going on there? What are they kind of, and how can they influence fatigue in any way if they do, and if it's meaningful?
Yes, so they have been our favorites in the last few years, the reactive oxygen nitrogen species. They're also called free radicals. And to prevent them from damaging things, you need antioxidants. And everyone basically knows that antioxidants are good for you. I mean, in the advertising on various, whatever it is, it's this thing contains a lot of antioxidants, which then indicates that this is good for you. You should really have it. But if you look more closely at it, so when you do heavy exercise, you will increase the production, if we can call them the free radicals, and they are potentially dangerous. So if you get a lot of them at the wrong place, or if you have a prolonged increase, they are increased, the production is increased all the time. They can go and damage proteins, membranes, DNA, and so on. But with exercise, they can also, when they increase during physiological kind of exercise, they will do things, but then one could say that they act like a very, very effective sensor or If you train endurance, for instance, there must be a way for the muscle fibers to understand that they have a problem. So you train, you cause a problem in the muscle fiber, they try to adapt, so the next time you do the same thing, they're better prepared. That's what training is all about, both with resistance and endurance training. And then you need some sensors. Someone has to tell the muscle fiber that it has a problem. And there the free radicals are, for endurance training, ideal. So they can go in and change, they can do small changes to the ryanodine receptor, and they can do other things. And in that way, the muscle fiber can sense that, oh, I have a problem, I have to do something. And what it then tries to do, if you do endurance training, is that it tries to be better on oxidative metabolism, and it also increases the antioxidant defense in its own or inside the muscle fiber. So from fatigue perspective, in our hands at least, the increased production of free radicals don't do anything really to fatigue development, but it's important during recovery. So it will slow down, if you have lots of them, it will slow down recovery. So full recovery can take 1, 2, 3 days in some cases. And that involves the free radicals. But at the same time, they trigger the adaptation, which is sort of the basis for the training. So what we have done is several different types of experiments, that if you have a high dose of supplementary antioxidants, and then you do some kind of endurance type training, we have looked at high intensity interval or sprint interval training, and then the effect is gone missing completely. So you can train and you have absolutely no beneficial effect if you take away the free radicals with antioxidants.
So free radicals are needed to an extent, but too much of them is essentially a bad thing. So there needs to be like
Yeah,
a balance,
exactly.
right?
So in the training context, they are your friends. They are makes it possible for the muscles fibers to adapt. In the pathology context, on the other hand, it can be too much of them over too long a time.
and so with fatigue, it's more of not of a cute thing, but it's more of a long, more of the long term game where the free radicals, as you're fatiguing, they're not necessarily contributing to that fatigue, but they start contributing to fatigue because over the course of hours or maybe days, they disrupt the cellular stuff that then affects fatigue.
Yeah, then can go in and yes, because that's something that I think everyone that trains hard have experienced, that after a really tough training session, the muscle fibers feels weaker for quite some time, maybe 1, 2 days or something like that, if you have done something really severe. And this prolonged version of lack of recovery, there the free radicals seem to have a central role, especially with endurance training. With resistance training, it's more that you get micro problems in the motor system, one would say. And that is in some way that all the details are definitely not known, but that also causes that you, after a really heavy exercise session, especially with eccentric, when you stretch the muscles with heavy weights, then it takes long to recover. A perfect way to adapt so that you get stronger, but the downside is that you have to spend some time doing nothing really to recover from it.
I see. So we don't really want to fully eliminate them, but they they seem to be the only mechanism to have the long term effect.
Yeah, the free radicals, yes, and I would stay away from excessive antioxidant supplementation. Luckily,
So,
many of the preparations you're supposed to take to live a happier and longer life, they contain basically nothing. The those doses are so small, so it doesn't really matter.
which doses? The ones that we see like in grocery stores with marketing, or?
Yeah, that kind, yes. But in the gym, of course, people can take a lot more. If you take, let's see if I remember now, vitamin C, I think it's 1 gram per day or something like that. Already then it can start to cause problems. And I'm pretty sure that there are recommendations out there that you take an awful lot more than 1 gram a day. Vitamin E is another very strong antioxidant that also can destroy the system. The problem with that you take it supplements is that you have inside the cells and inside your body, you have very sophisticated systems to take care of these potentially dangerous molecules. And you disturb the system if you take it if you take supplements. So then the guys that are there to do the job and are very good at it, they can't operate anymore because the situation or the environment has changed.
So that one's interesting then, because you always see in the grocery stores where you see like a lot of marketings in just antioxidant drinks where they just ramped up the prices and said that these are super healthy for you, and then the gym people want to start taking them because they're healthier, but the doses that are in those, I guess, grocery store drinks aren't really a lot, so it's not worth it. And then we don't even really want to drink a lot of doses because then that reduces the benefit of the free radicals.
Yes, I think it's money down the drain. And I think many of the products you see in normally grocery stores, if they contain high enough concentrations of antioxidants and so on, then at least in the Swedish environment, you can't sell them because you can't sell things that are, you can't sell things in normal grocery stores that are supposed to have evidence of real pharmacological effects. You really must prove that they're useless before you can sell them if one stretches it a bit.
So, I know some of those companies have like their own studies and research groups, but I think those are very biased, I believe.
Yeah. And this piece of cake to show that free radicals are dangerous. There are many non-physiological ways of doing experiments to show that they're really dangerous, and then you can put on the antioxidant and see that things improve.
And because it's so easy to show that they're dangerous, that's how they market it, and then they could sell it.
Yeah, I guess so, yes.
Interesting.
So experiments like you have, if you do cellular experiment, you have some cellular experiments, you put on some free radicals, the cell dies or the cells die in 30 minutes, say, and then you put on antioxidants and they don't die. And then you have to show that it's effective. It's only that, luckily, when we do a training session, cells don't die after 30 minutes. It's sort of just a perfect or normal challenge to them.
So, I think that was the last mechanism with peripheral fatigue, which I guess that led more into like muscle damage a little bit rather than acute peripheral fatigue with the free radicals. But one common thing that I'm hearing right now is that the production metabolites like inorganic phosphate or the other factors of peripheral fatigue that we talked about, they're stating that in vivo or in resistance training, they're not necessarily causing large decreases in force, and that's mostly over-exaggerated by the research, and it's mainly a reduction in shortening velocity of the muscle fibers, but the force production of the muscle fibers doesn't really get impacted by the buildup of metabolites like inorganic phosphate. I wanted to get your opinion on that because I was always a big believer that I think, accumulation of inorganic phosphate can reduce muscular force output in vivo.
Yes, so I think it's might be a bit difficult to explain, but how do you define force and so on and speed? So I imagine if you do something with heavy weights, the first reps, it's easy, you can lift them rapidly. And then it becomes slower and slower, your ability to lift it. And then you think that the muscle then clearly becomes slower at doing what you want to do. But that's not necessarily because of the muscle fibers getting slower. It could be that they become weaker. And when they're weaker, they have to work at a higher proportion of their available force, which means that their shortening speed goes down. I don't know if that's understandable, but actually they look slower because they're weaker.
Let me give an example, because this is actually something that we also wanted to get into before we end this podcast. It was the idea of the force velocity relationship in training, where people believe that, let's say you get a weight again during a bicep curl, and you're curling for 8 reps, maybe 10. The first few reps have this relatively consistent speed, maybe a little faster. But as you get near the end, where you fail on the 8th or 10th rep, your velocity or your shortening speed starts getting slower. You can see people getting slower as they start trying to curl. Now, because people think that the force velocity relationship is that the slower we move, the higher force we produce, like, well, because we're curling the dumbbells slower unintentionally, then that means fiber force is increasing, right?
Mm.
What do you think of that reasoning?
No, I don't think that is not really correct. You should, I think one can explain it with power. That to lift something, you need a certain power, and that is the speed times the force. And then when the muscle becomes weaker, which is dominating, so if you have the force velocity curve, the function goes down a lot more on the force axis than on the shortening velocity axis. But you still need to produce the same power. So when force is going down, then you have to use a much higher proportion of the available force. So you have to move up on the force velocity curve, so to speak. So it looks like, or move down on it, so it looks like they become slower, but they are slower because they're weaker. It's difficult without showing a diagram to show how it must be like that, but
So essentially, it's more of a consequence of the muscle fibers getting weaker that we become slower.
yes.
Okay, so it's yeah, because many people say, like, as you move slower, you're increasing force. But I think those force velocity diagrams are showed like in unfatigued conditions, and now when we're in resistance training, where we have fatigue conditionings happening.
Yeah, exactly. So towards the end of when you can't really do more reps, then if you have a force velocity curve, you have to move the curve down on both axes. And you move it down more on the force than on the velocity axis. And then you have a new curve. And then you have to try to, from that curve, produce enough power to lift the weight. And then you will see that the velocity will be a lot lower.
So I'd like to finish off with 2 more questions. With the first one being, then what kind of causes the shortening velocity? Then is it a mixture of all of the metabolites? Is it mainly hydrogen ions? But you mentioned that hydrogen ions aren't probably that important, and it could just be the inorganic phosphate that's decreasing the cycling rate of the motors, or what could be happening there that's causing the shortening velocity.
So if you mean pure effects on, we forget about force for the time being and just the shortening velocity. then most likely it's the hydrogen ions that will cause some slowing. And then when you have you're approaching exhaustion, so to speak, then you can have ADP, adenosine diphosphate spikes during the contraction, and they effectively also slow the cross-bridge cycling. So early on you can have the hydrogen ions, and later on you can also get the ADP transients.
But inorganic phosphate doesn't slow velocity, but it does affect the ATP, which can slow down the velocity, right? So it kind of indirectly slows down velocity.
Yeah, so phosphate ions as such don't do it, no. They reduce on the force velocity curve, they reduce the force on the force axis, but they don't do anything to the velocity axis.
Not,
But
not
as
it.
we discussed before, when force goes down, then the contraction as such becomes slower because you're now at you need to activate more of the available force.
So they can reduce force indirectly, right?
Yeah, they can do during a normal exercise they can reduce the speed indirectly. Yes, they reduce the force, which could lead to reduced speed indirectly. If you look
So
at the end of a 400-meter run, for instance, most runners run a lot slower there. And I think there is, you really have the phosphate problem at its maximum there. So they don't, they, to a large extent, they run slower because they don't have enough power or force to do fast running anymore.
So, I mean, in a practical scenario, whether you're in a gym or you're running, and you start decreasing, shortening velocity, that is a mixture of the hydrogen ions directly decreasing the speed, but also the other factors of peripheral fatigue that are reducing force output, which is also feeding the decreased velocity. And it's really
Exactly.
hard to tell which one is doing more of the work, but at the end of the day, we just know it's both.
Yeah, one could say so.
Okay. And so, I mean, I guess we could finish off with the clearing up of metabolites once these fatigue mechanisms are coming into play. Do you know if there's like a certain time frame on how quickly they can clear up, or how quickly inorganic phosphate or acidosis can, uh, yeah, clear it from the muscle in a way? Does it take minutes, hours, seconds?
Minutes. So the phosphate ions, they aren't really cleared from the muscle. So at rest, they are the majority of them are bound to creatine in the form of creatine phosphate, our fastest energy buffer. Then when we stop exercising, you take the creatine ions that are still there and you take the phosphate ions and you provide energy and then you make new creatine phosphate. And that takes minutes. Severe depletion, I would say that half an hour or something like that, the creatine phosphate stores are there again. And you can, I mean, in from a really simple perspective, you can see after exercise, you breathe a lot, which means that you need a lot of aerobic metabolism. And aerobic metabolism is what's building up the creatine phosphate stores. So after exercise, you breathe more heavily for some minutes, and during those minutes, you have a rapid recovery of the creatine phosphate stores.
So is it directly related to our respiratory work? Kind of like if uh, if we're breathing heavy, then they're clearing up. But as soon as our breath kind of goes back to resting, that can be a good indicator to show that our metabolites are fully cleared out, or could they still be lingering around?
Yeah. The most acute part of it has when you stop sensing that you breathe a lot more heavy than you did before exercise. You can say that well now my creatine phosphate stores are not fully rebuilt, but they are on their way. And also it goes with endurance training. It becomes faster. So you have a higher aerobic capacity, which means that you can build up the creatine phosphate stores more rapidly.
And yeah, and then you see the runners kind of breathe heavily for a minute, and then they could start running again once once their breath or their breathing kind of becomes back to normal ranges, right?
Yes, exactly. Then one can say that all the consequences of the first bout of running uh, that required anaerobic metabolism have now been restored. So now you're back again, and then you can do another anaerobic session. You have a built up and changes of metabolites, and then you breathe afterwards, so to speak, to build them up again.
And I think that takes anywhere from like 3 to 5 minutes, maybe around there, depending on the intensity and work you put into it, right?
Yeah, and the duration, if you have a completely depleted stores, it of course takes longer.
Now, what I find interesting is that in resistance training, um, when you start off, you're good. Your breathing is normal, and then as you kind of go on into the gym, your breathing is still normal, but you have uh, like a pump going on, or like your muscles are getting more exhausted. Could that indicate that like the fatigue mechanisms are still there despite your breathing being back to normal?
Yeah, yes, yes, of course. I mean, if you still sense that the muscles are weaker or they all haven't fully recovered and you're breathing in normal, yeah, of course.
Because
And
I
it
think
could be that it's a type of exercise that is not really cause fatigue from a sort of a metabolic, aerobic, anaerobic perspective. It's another type of exercise.
okay, so with because I think people might take this as like, well, breathing is a direct indicator that metabolites are cleared up, and as you said, it kind of is, right?
It kind of is, but it's not perfect in any means. It's the grayscales to it.
Okay, yeah, so it's like a little indicator. So yeah,
Yeah,
just in case people don't go out and start saying like, oh, I'm breathing normally again, so I'm all good. But I think acutely it could be a nice indicator. But of course, if you keep doing work, then I think the separation could increase,
yes, yes,
and
and then
everyone's
depend on what you do. If you do some kind of exercise with 3 repetitions, and then you wait, and then three, and not, then you don't. It's not really the metabolic kind of fatigue. There are other things that come into play.
absolutely. All right. Well, I think that kind of covered everything I wanted to go over today. I will, for people who are interested in continuing to read more into this, I will link your new paper down below, and I think that's where you go more in detail with everything in the literature. And of course, it is a big read, but I think it's worth your while. Dr. Westerblad, thank you again for joining us in this episode. And yeah, I think many people will find it very valuable. So, thank you guys for listening, and I'll see you guys on the next episode.
Okay, fun to be here. Thank you.